Hill is a professor in the departments of cell biology and anatomy and psychiatry at the Hotchkiss Brain Institute at the University of Calgary and a Tier 2 Canada Research Chair in the neurobiology of stress. Dr. Hill is the Executive Director for the Canadian Consortium for the Investigation of Cannabinoids and the past chair of the Gordon Research Conference on Cannabinoids in the CNS and past president of the International Cannabinoid Research Society. Dr. Hill's research has primarily focused on endocannabinoid signaling, its dynamic regulation by stress, and how it modulates neural circuits governing stress, fear, and anxiety. More recently, his lab has begun to investigate the impacts of developmental exposure to cannabis using novel vapor delivery technology for rodents. He has published over 150 scientific papers in the field, has sat as guest editor for several scientific journals, and regularly consults for both industry and governmental agencies. Dr. Hill has been the recipient of the Young Investigator of the Year Award from the Canadian Association for Neuroscience, 2016, International Association for Cannabis Medicines, 2017, and the International Cannabinoid Research Society, 2018, and has been elected as a member of the Royal Society of Canada College of Young Scholars. Dr. Hill has regular engagement with the media and has been featured in the New Yorker, New York Times, Scientific American, Vice News, CBC News and Radio, as well as in a documentary on cannabis produced by PBS Nova. So without further ado, please join me in welcoming Dr. Hill for his talk.
Thanks a lot, Lonnie. It's a pleasure to be here, and I'm kind of happy to talk about the stuff. This is admittedly a relatively new area for me, I will admit, in the sense that I'm not a neurodevelopmental scientist at all. Like Lonnie had said, my background and research typically focuses on stress and anxiety, although with changes in legalization and moving into the cannabis field, I actually became very interested in a lot of this stuff. And so that is why we've started to move into this. So I think the way that I'm going to go through this today is I'm going to just start with kind of a general overview of kind of what we know about endocannabinoids and brain development, and then go into a little bit of what we understand from the human data about cannabis exposure in humans. And Lonnie actually has already covered some of this, and her stuff itself is actually quite interesting developments in the area. And then I'm going to end by kind of going into some of the stuff that we've been doing in my lab, the approach we're taking, and kind of where I think some of the interesting stuff in the field is going and things to chase. So, okay, so the first thing is to kind of introduce the endocannabinoid system. I mean, this is something I think at this point that most people in neuroscience are generally familiar with. This is kind of the canonical view of how a lot of us view endocannabinoid function. It's typically viewed as a retrograde signaling system in a kind of adult mature central nervous system where a postsynaptic neuron becomes activated by something like glutamate that's released from a presynaptic terminal, and this triggers a series of biochemical events that causes the synthesis and release of endocannabinoids like anandamide or 2-arachidonoglycerol, which can be seen over here. They're both kind of arachidonic acid molecules coupled to either an ethanolamine or a glycerol tail, making them kind of a very interesting, unique class of signaling lipids in the brain. As retrograde signals, these molecules signal backwards. They cross back across the synaptic cleft in a manner, actually, I would have to say we still don't truly fully understand, given that they're lipids moving through an aqueous environment. And they activate presynaptic CB1 receptors, where in the canonical view of endocannabinoid function, their main role is to really inhibit calcium influx and largely through a few other pathways as well, suppress neurotransmitter release. So we often look at this as kind of a circuit breaker system. When a neuron becomes too activated, it releases endocannabinoids, it feeds back, turns off afferent input to kind of reduce the amount of excitatory transmitter release and keep things in check. There's also a lot of heterosynaptic effects in other systems that really ultimately work to guide the balance of synaptic plasticity and changes in a lot of different kind of short-term and long-term plastic phenomena in the brain to keep circuits behaving the way they should optimally. Now, that's always the way that we think of endocannabinoids, and this was a figure Lonnie had in her talk. I'll focus on it a little bit more just to kind of go through this a bit more. But one thing that we understand is endocannabinoid function in the developing brain, especially in the prenatal embryonic brain, is a fundamentally different process than it is in the mature adult or even early postnatal brain, when it really kind of takes on the synaptic role that it does in the adult brain as is. And as Lonnie had pointed out, what we see is this kind of really interesting dynamic change in endocannabinoid function across the gestational window. And the thing I have to say, there's a lot about this that is very fascinating and kind of unique. So one thing is, it is almost mind-boggling to me to think of all the different levels during developmental processes that endocannabinoids have the ability to influence. And there's a fair amount of work showing that they play a role in fertilization and implantation of the blastocyst, and that endocannabinoids are required for these processes. And then once we actually get embryonic development going and neurodevelopment starts to kick in, we start to see different and more pronounced roles of endocannabinoids, and they're important for a lot of basic processes such as neurogenesis, or even regulating the quiescent state of progenitor cells and whether they go into a proliferation state. There's a big effect of endocannabinoids on fate determination, and a lot of work suggests that they also promote gliogenesis in this capacity. There's documented effects of endocannabinoids on radial glia, and showing how that they're going to affect the kind of association of how these cells move as they go into development of various columns in the brain. And importantly, one of the things that I'm going to focus a little bit more on today is this role that they play in axon guidance. And I'll go into this in a little bit more depth. And it's also important to note kind of this image across the top, which is what Lonnie was pointing out as well, which is this kind of differential effect that happens with endocannabinoids across development. And one of the things that's really interesting is these early phases that are really non-neuronal in nature. They're really more about regulating the egg and the blastocyst and the fertilization and implantation stages. These seem to predominantly be driven by the endocannabinoid anandamide. And this is the period when anandamide levels are high. And once implantation occurs, anandamide levels actually go down a fair amount. And then what happens is 2-AG levels start rising. So there's almost this kind of trade-off that occurs between the two endocannabinoids, where 2-AG seems to become, from all intents and purposes, from the work we've seen, 2-AG becomes the primary endocannabinoid in this period of neurodevelopment, which is more involved in kind of axon guidance. And then once we kind of shift into the adult state, obviously both endocannabinoids seem to play a role in a lot of processes, and they do still seem to regulate things like gliogenesis and neurogenesis, but more importantly, they're regulators of synaptic transmission. So to kind of go into a bit more depth about the axon guidance stuff, because I'm going to focus on this, because for one, this is actually a process that has been worked out to, I mean, I would almost say immaculate detail. It's almost shocking to me that the researchers, primarily people like Tibor Harkany and Yasmin Hurd, who've done a lot of the work in this area, have been able to create such a detailed analysis of how the system is working during the axon guidance phase. And so this is kind of a crude picture of what we see with a developing neuron, where we have what will ultimately become the presynapse, but is the axon that's kind of moving to form a new circuit. And so a lot of this is based on cortical neurons, corticothalamic, corticofugal neurons that seem to go undergoing development, where the axon extends out. And in this situation, what we actually see is going on is that there is a clustering of this enzyme called diacylglycerol, which is responsible for the synthesis of 2-AG, which is an endocannabinoid, as I mentioned. And what actually seems to be happening is, in this situation, 2-AG is almost functioning as an autocrine signal, where this part of the axon head that's moving to find its synaptic contact and form the circuit that will ultimately be its final target, it uses this autocrine ability where the actual cell itself synthesizes and releases 2-AG, which almost acts as some kind of a chemoattractant signal to drive where the axon goes. And what's really fascinating about this process is the entire neuron does seem to possess the ability to generate 2-AG, but to maintain the specificity of where the axon guidance is heading and the direction of the growth, the neuron actually develops this ability where, as you can see in pink here, there is a massive clustering of an enzyme called monoacylglycerol lipase, which is really there to chew up and degrade 2-AG. And what happens is this part of the neuron has a massive clustering of MEG lipase. And what this does is this acts as almost a rate limiting step for 2-AG signaling. And so what it does is it quenches 2-AG as it's produced by diacylglycerol, therefore preventing it from actually acting on CB1 receptors. And the point of this is, because 2-AG is kind of a chemoattractant signal that drives axon guidance, what the cell does not want is an inappropriate signal being generated to cause this part of the cell to start branching off and going in different directions. And this is a really, really molecular specific mechanism that a neuron develops during this part of development to maintain the specificity of where this 2-AG signal is, so that it concentrates explicitly on the axon head, so that's where it's going when it guides. This is a fascinating process, because then what happens is, once the neuron finds its target, the way that it finds its target, if we focus here on the left, this is the immature neuron in a different representation. And what we can see again is the growth cone component, then what will become the presynapse, is very rich in diacylglycerol. So it's making all this 2-AG to drive it in an autocrine manner. And then you have this part of the stabilized axon, which is mag lipase rich to prevent 2AG signaling from causing misdirected cue guidance for that growth cone to go towards. As soon as that neuron finds its target where it's supposed to go, that postsynaptic target attracts the growth cone by itself releasing 2AG. As we can see, there is these domains that exist on the dendritic components of a cell that have 2AG, and they make 2AG. So this is the part that the neuron starts becoming attracted to. Once that growth cone finds its ultimate target and it determines where it's going to go for its fate, let's say, so this is going to be the circuit that it will mature and become a part of, the neuron rapidly polarizes and undergoes this massive shift where suddenly the axon head, which was the growth cone, forms into the presynapse, and diacylglycerol essentially flushes out of this part of the neuron, clusters into the somatodendritic region, and then this whole part of the axon becomes very rich in mag lipase. And now we have a completely different molecular structure of how the neuron is assembled in terms of the components of the endocannabinoid system, because now it is set up to appropriately function as a retrograde signaling system where you have the cell body, or as we can see over here, this dendritic spine, which is where 2AG is now made, and it will be released and then act presynaptically onto these receptors on presynaptic targets. So this is really an amazing process, and it's not entirely understood the mechanisms that will cause this polarization of where diacylglycerol suddenly shifts once a synaptic target is found. But I think the important point to recognize is this is a really important growth signal. It's very important as a guidance cue, and it's very important for the appropriate maturation and functioning of how a circuit is going to assemble itself, find its appropriate synaptic target and end. And we can understand how important this is because of the fact that the cell has developed this very specific organization of where these proteins end up clustering, such that we have this portion to prevent misguided cues being rich in mag lipase to prevent excess 2AG signaling, and this part explicitly in the growth cone to be where 2AG is made to drive the signal. So we can understand, obviously, there's a necessity of this level of specificity. So this then starts to make us question the fact, okay, so what happens then when cannabinoids are introduced to a developing brain? Because we have a very tightly regulated and a very spatial and temporally specific system that dictates where neurons that are undergoing this neurodevelopmental process should be going. And so one thing that we know, certainly from in vitro work that has occurred in slice physiology preparations, where we've actually, or others, the field has started looking at this, it's become very clear that due to the pharmacological differences that exist between agonists at the cannabinoid receptor, like THC from cannabis, or 2AG, is that they actually are not very similar in a lot of the ways that they signal. And so one thing that we understand is 2AG, for example, is what we would refer to as a full agonist. So when it binds to the CB1, it's very efficacious at recruiting G protein signaling, and it mounts a very robust intracellular response. THC, on the other hand, is actually really only about a partial agonist in terms of its ability to signal at CB1, but it has an incredibly high binding affinity, much higher than 2AG. And what we've learned from in vitro preps from animals that are kind of pubertal or older is that because of the high binding affinity of THC versus 2AG, it actually has the ability to almost act as a functional antagonist in some ways, or at least an occluding signal, because what it will do is it will pre-saturate CB1 receptors and prevent 2AG from doing its thing. And so if we start to think about it in this context, and we really start recognizing the fact that the way that the biochemistry and molecular organization of these developing neurons are, they're meant to be in a very specific fashion. As we can see right here, normally 2AG wouldn't be signaling at this receptor, but suddenly when THC is introduced into the system, we've lost all our specificity temporally and spatially. And so what ends up happening is that THC will just indiscriminately activate CB1 receptors, and therefore we've lost all this very fine-tuned specificity that exists. So now I do a lot of kind of talks to the general public, and I'm someone who always likes to think in analogies. And so I always try and think like, what's a good way of analogizing this? And so the analogy that I've come up with about the impacts that this could have, and I emphasize the word could because there's such a paucity of data for the most part in this area. A lot of this is assumptions that we've made based on the biochemistry, but I think a very reasonable assumption to be made is what this is happening in terms of how this could influence the specificity of the guidance cues. And so what this made me think of in an analogy sense is actually something I learned when I was on vacation years ago, when traveling was actually a thing down in the Mediterranean in Turkey, which was the impact of nesting baby turtles. And this may sound weird at first, but basically what we've learned is that baby turtles, when they're born on the beach, they have a natural affinity and are attracted to light because the moon will reflect off of the water. And so for them to be able to move from hatchlings and go into the water, they naturally follow sources of light because that is usually indicative of bodies of water for them to move to. And so this is the directionality of how baby turtles normally move. However, with the development of all these seaside resorts that have happened, we have all this artificial light that's developed. And what's ended up happening is that this can overwhelm the light produced by the moon, and it actually acts as a false guidance cue. And so what ends up happening with a lot of these turtles they found is that they begin to follow these light cues as opposed to the real light cues. And it ends up creating a different fate for them, where they go in a different direction than where they should be. And for a lot of them, it actually ends up resulting in death because they dehydrate because they're not going into the water or not in the right environment. And so this is what I think of when I think of what THC could be doing. To me, THC is kind of like the resort lights on the beach, in the sense that it has the potential to be acting as a false guidance cue to drive the development of a circuit to go into a differential fate than it probably should be, because of the fact that we've lost the innate natural cue that really should be there. So that's my analogy for the day. Hopefully that resonates with some people. Okay, so then the question really comes about, this is all speculative because this is based on what we understand about the biochemistry. So really, what do we understand about exposure to cannabis during these kind of sensitive neurodevelopmental periods in humans? Now, as one can imagine, this is not the easiest thing to study. And I think it's an important point, as Lonnie had pointed out in her talk, is, well, what is the basis? Is there something to be concerned about here? Like, how frequent is this? Now, as Lonnie had mentioned, the data here is actually quite patchy and for multiple reasons, difficult to obtain accurately. Now, one thing that we can see is that, at least in these kind of data that have been collected in Canada, and this was pre-legalization, so there's going to be more updated data that comes out soon, as we'll have greater understandings of the impact legalization has. But although we can look to some of the United States that have gone legal before us to look at this, but we do see increases in cannabis use among pregnant women. And what's interesting is it does tend to be more prevalent in younger women who are pregnant, that they see higher rates of cannabis use in. Now, these numbers do tend to suggest that we're looking at least for women under the age of 24, between 6 and 7% are using cannabis during pregnancy. There is actually some data that's come out of Washington State looking at umbilical cord blood and doing toxicology at it that would suggest in this age range, it may actually be close to 20% of women who are using cannabis. So it's difficult to understand the magnitude of this effect, but I would definitely not say it's a trivial effect. It's something that, if there is effects of this, it's going to have a relatively large effect in terms of gross numbers of people. Now, one thing I think we can safely say at this point is cannabis is not alcohol. In terms of its teratological effects and its potential neurodevelopmental effects, it doesn't seem to produce the same host of issues that have been seen with in utero exposure to alcohol. Certainly, we don't seem to see the facial dysmorphologies that are kind of present in alcohol. Now, it's interesting because we do know that a lot of those effects from alcohol are driven by interactions with sonic hedgehog system. And there was some recent work that's come out of UNC down in the States that would suggest that synthetic cannabinoids may actually have the ability to do this. And again, this is a point I'll come back to in terms of how we model approaches to cannabis use in humans, because these molecules do behave a bit differently. And there's also some work that suggests co-administration of THC with alcohol could enhance some of the teratogenic effects of alcohol itself. So there are more complex interactions here at play that we also should consider. But what do we actually know about this? We know that it doesn't seem to produce a gross immediate phenotype that's as obvious as the effects that we see from alcohol. So what do we actually know, and how do we know this? So the first thing that we can look at is kind of some neonatal outcomes that have been associated with maternal cannabis use. And Lonnie had mentioned that a lot of the data is very patchy and inconsistent, and that's an understatement, I would say. I would say there is large inconsistencies that exist in the literature. And as we'll see as we go through where the data comes from, it's not surprising why we're seeing this. But basically, I would say when we look at neonatal outcomes, one of the most reliable effects that is seen from prenatal cannabis exposure is a higher percentage of infants that come in at low birth weight, which is determined as 5.5 pounds or less. There does seem to be a reliable effect of about 100-gram difference in birth weight from infants exposed to cannabis versus those that not. Now, some of this may be driven by tobacco use. And again, this is one of the complications because cannabis users tend to, depending on where you are, having grown up in British Columbia, I would make the argument you could probably find a population in British Columbia that uses cannabis and not many other substances, but typically elsewhere in the group that are using cannabis, you often are seeing co-use of tobacco, possibly co-use of alcohol and other substances. And so trying to control for these can be difficult. However, certainly when tobacco is controlled for, some of these effects do disappear or dilute significantly. There is limited evidence of an association between maternal cannabis use and NICU admission. And interestingly, there was really no strong evidences for differences in birth length, head circumference, intrauterine growth restriction, or congenital malformation from maternal cannabis use. And this is based on a meta-analysis that came out in 2016. So that's the kind of neonatal outcomes. And really the one that kind of jumps out reliably is the lower birth weight. There doesn't seem to be a lot of other effects that have come out from this. So what about longitudinal studies? So there have been 3 real cohort studies that have been done to date that have really informed this research body. The first one was a cohort that started in the 70s that Peter Fried, who was actually in Ottawa, organized and was the one who tracked the individuals and the children as they grew up. This cohort is an interesting cohort. It was in the 70s. So for one, the cannabis is going to be, let's say, definitely have a different flavor than modern cannabis because it would have been much lower THC. The cohort of women who would have been in this study, the descriptions I've heard is some of them are kind of like barefoot earth mothers, probably relatively decent SES levels. So we're not talking about massive poverty stricken. It's unclear about the details of some of the other aspects. The MHPCD cohort is actually fundamentally very different in nature because it's driven, I believe it's from Baltimore to Detroit, but it's a high risk cohort. That lives in low SES with a lot of external stress in their life. And then the Generation R study is one that was based out of the Netherlands that started in the early 2000s. So it's kind of a more modern one. And again, Netherlands, because of the decriminalization of cannabis and the use, that's one that they've kind of capitalized on the fact that they had access to populations that would be more open about the fact that they had used during pregnancy and tracked them. So those are kind of the 3 cohorts where a lot of the data that we understand has been coming from. Now, again, as I will review in a second, these are kind of findings that have come from some of the individual studies. And the reality is that the aggregated data does not hold that well. And some of that can be explained by the differences in the cohorts between them. But in general, these are the effects that we start to see coming out in these cohorts in an age-dependent manner. You do tend to see poor scores of memory and verbal measures around age 6. Around age 10, there is some evidence that they may see more hyperactivity, impulsivity, inattention. There's other effects on impulse control and some higher cognitive effects in visual reasoning, visual problem solving around 9 to 12. Once we get into early puberty and adolescence, we're starting to see decreased attentional stability, some possible delinquent behavior, although there has been some factors that have contributed to that. And then as we move into young adulthood, doing neuroimaging, there's been some suggestion of increased PFC activation, poor response inhibition, and a go-no-go task, possibly increased risk of using tobacco and cannabis, although even in the Ottawa cohort study, there's been some discussion about what could be mediating this because some of the discussions surround around the fact if you have a mother who is openly smoking pot during pregnancy, the kind of general attitudes about drug use in that household may be very different than in one where that was not the case. And that in turn may have influences on subsequent drug use of the offspring that could be independent of any neurobiological effects. So disentangling social and familial aspects from biological effects can be tricky in some of these situations. And that certainly is where the advantage of animal models come into play. But it's kind of interesting to kind of look at this, that there are some signals that have popped out. Now, a few years ago, the National Academy of Medicine in the United States did a very intensive and massive analysis of everything that we understood about cannabis, both on the potential health disease treatments that it could be used for from a health perspective, but also the harms that are associated with cannabis. And this was based on a review of every published study that had basically been done to date. And they kind of took the data together and weighted the outcomes and the reliability of the findings and created some general conclusions that can be made. And when we look at the prenatal and perinatal and neonatal exposure periods, again, as I had mentioned before, the single variable that seemed to be reliable that came out of this was lower birth weight of the offspring. So that does seem to be, I would probably argue, the most consistent effect of prenatal cannabis exposure. There is limited evidence of a statistical association between maternal cannabis use and some pregnancy complications for the mother and some admission of the infant to the neonatal, the NICU. However, looking at the meta-analysis that came out around the same time, this was highly inconsistent, and that is why the National Academy in review kind of put this down under limited evidence. And they made the conclusion ultimately that despite some of the things I showed you in the previous slide, that there is actually insufficient evidence to support or refute these claims about the associations between maternal cannabis use and later outcomes. This includes things in the early children, such as SIDS, but also some of the more later effects that I was referring to, like cognitive or academic effects, or later substance use, like the subsequent use of tobacco, cannabis, or opiates, which is one of the effects that has come out of some of Yasmin Hurd's work. So really, we aren't really in the most informative place right now in terms of what we really understand about this. And so I think what we can say is that it doesn't look like prenatal cannabis has what I would call perhaps sledgehammer effects on the fetus, the same way we've seen with alcohol exposure. But given what we understand about the importance of endocannabinoids during neurodevelopment, it would be quite surprising to assume that this would have no impacts whatsoever. And so likely where most of the field has fallen, I would say, is this kind of decision that most likely what's happening is there's probably some aberrancies in normative neurodevelopment occurring, but they may not be immediately apparent. They may come on later in life, or as has been discussed already at this meeting, there may be the requirement of kind of a second hit model. So it may just be that there's what we would refer to as like a latent effect, that there could be something there that might enhance the risk of vulnerability to develop a mental illness or a physical condition in some capacity, but it requires a secondary exposure to another challenge like stress or inflammation or some other environmental agent that would bring the latent variable out. And so those are the things that we're kind of unclear of as of now. So more recently, there's been some other data that has started to actually look at associations of prenatal cannabis exposure. And these have been done in large population studies. And this is where I start to actually find things that are some very interesting signals that are starting to come out that I think, in my view, is really where the field should be moving to some degree. So one of them was a 2020 Nature Medicine paper that came out from Ontario. And they did a large scale data analysis looking at a carefully matched sample of prenatal exposure to cannabis to not. And really what they found was that there was a significant association for an increased risk associated with the development of autism spectrum disorders, or ASD, in children that had been exposed to prenatal cannabis. Now, while this isn't accounted for here, one of the things that's well established is that a lot of neurodevelopmental disorders, such as autism, tend to be more prevalent in males and females. And I'm going to come back to that because I think that this is something that is one of these interesting effects that's coming out. So this is the first large-scale study that I think has shown an interesting relationship between a neurological or neurodevelopmental disease and prenatal exposure to cannabis. Similarly, researchers, now that there's been enough kind of data generated from the ABCD cohort studies that have been going on in the States, they've been able to start looking at some of the variables here. So one of them, this was looking at going back through the cohort that they're looking at and studying the individuals who they knew had exposure to cannabis during pregnancy. Now, and Lonnie had mentioned this before as well, and this is an important point, is that the timing of when the women are using cannabis during gestation is obviously a critical aspect of this. Now, the way that the ABCD study has broken this down is they had prenatal cannabis exposure prior to maternal knowledge of pregnancy, which would usually suggest this is at least the first half of the first trimester, perhaps a larger proportion of the first trimester, depending on exactly when they found out they were pregnant. But that's going to be focusing on the first trimester. And that's consistent with the levels of use, as Lonnie had pointed out, which is also due to the fact that the primary reason that women report using cannabis during pregnancy is to maintain morning sickness, which is far more prevalent during the first trimester. So there is a lot of suggestion that some of these effects may relate to first trimester exposure. And so what's interesting here is that this effect was specific to prenatal cannabis exposure that occurred after maternal knowledge of pregnancy. So this means that the mother knew she was pregnant and continued using cannabis. So now we're looking at exposure that spans a larger period of the gestational window, if not the whole thing. And so I'll come back to that with the next slide because there is differences in terms of the duration of when they're exposed. But what this study found from mining the ABCD data was that it found that in children who had been exposed to cannabis for likely the large majority of the gestational window, there was this significant increase in proneness to develop symptoms that are psychosis proneness. It's kind of like a prodromal questionnaire that gauges various things about thinking patterns and establishing pre-psychotic level behaviors. And so what they associated was there was this relationship with gestational exposure, with psychosis proneness, and kind of diffuse psychopathology during childhood. So again, this is interesting because again, we're still dealing with a schizophrenia now, theoretically, if that's what this results in a conversion to, and we'll have to wait a few years for the data to develop more to see if this psychosis proneness does indeed equate to increased levels of schizophrenia. But again, like autism, we're talking about a neurodevelopmental disease. And I think that this is really key to this. So more recently, a collaborator of mine, Hilary Marusak, who is down at Wayne State University, she has also been doing some work looking at the ABCD cohorts on children who were exposed to prenatal cannabis. Now, here, what they found was they actually looked at white matter tracts, and they were looking at MRI data from the ABCD cohort, and they were focusing on frontal limbic white matter pathways. Now, this scans are done from children who are about 9 to 10 years old. And what's interesting in this one is that the effect that they see relates specifically to prenatal cannabis exposure before knowledge of pregnancy. So similar to what Lonnie was talking about earlier, this is the early period. This is probably more of a first trimester level of exposure that we're talking about here. But what they were able to detect here is that in the fornix, they were able to find small but significant reductions in the fractional Catherine, I'm going to mispronounce this. We'll say the FA signal. So the white matter signal here seems to be reduced. Now, I find this personally quite interesting because, again, one, I was talking about the neurodevelopmental effects of cannabinoids. We're talking about effects on axon guidance and circuit formation. And so this, in theory, in my mind, it makes sense that one of the areas that we may predict that we would see differences in would be white matter. So this is very early data. My lab's actually working with Hilary right now to do a comparable study in rodents and using MRI approaches similar to what Lonnie was talking about that they're doing, but focusing on white matter levels. This is one of the things that we're looking at right now to see, can we see when we have an animal model where we're able to control for a lot of the other variables, do we still see this effective prenatal cannabis exposure on white matter tracts, especially in the fornix in the brain. And so this is kind of hot off the presses data. It's unpublished. It's very fresh. Hilary just sent this to me last night. So I think this is really interesting. And this is kind of one of the things where I think the area needs to start going and looking more is a lot more, because of the effects that we know relating to axon guidance and circuit formation, I would predict that white matter would actually be an important thing to focus on. Okay, so as we can see, the data that exists from the human populations is relatively limited. I think that's fair to say. There's not a lot there. There's a lot of potential confounds in that data set. Because of all the other variables that come into play. Some of the cohort studies are so different in nature that it's almost difficult to actually collapse the data together to really look at it. And so given what we know about the biology of the cannabinoid system and our predictions about what could happen, really, this is an area that actually, to me, just screams for preclinical animal work to come in and really help resolve some of the questions. And I have to say how surprising it is that up until relatively recently, this is an area that was just not well explored. I don't think it was something that people viewed as a public health issue. I don't think it was something that people viewed as a health priority, certainly for places in the States like NIDA. They seem to be much more focused on adolescent exposure as opposed to prenatal exposure. So that was where more of the money was going. And so I think there are just honestly a lot of large knowledge gaps that exist here. So preclinical studies are obviously have a huge amount of advantages, but there's also some issues, again, similar to the human studies, that I really think that we need to think long and hard about in terms of how we understand this data. So the first one is, and this is something, I mean, I'm guilty of historically, and I think a lot of people in the cannabinoid field are, is that synthetic cannabinoids are not THC. We often used to just think of things as, oh, it's a cannabinoid agonist. So if we give something like WIN or CP or Hu210 or one of these other drugs, it's going to give us information that is going to be informative to the effects of THC. What we have actually learned in the last few years is this is definitely not true. I mean, first point is what I mentioned before, which is the efficacy of the agonist. THC we know is a partial agonist. So the way that it interacts with CB1 and it mobilizes intracellular signaling pathways is fundamentally different than all the synthetic cannabinoids. They are almost all full agonists. They all evoke very robust signaling pathways. But we also now know in the age of biased agonism that it's not just differences in the strength and efficacy of the molecule. They actually, we now know, recruit different signaling pathways internally. And so THC and a lot of the synthetics don't have comparable effects on their ability to recruit G proteins versus beta-arrestin. And so there's a lot of different effects that are produced by synthetic cannabinoids that are not seen in THC. This has also become very apparent in humans because as we've moved into an era where synthetic cannabinoids are actually being used recreationally, we've seen a significant number of fatalities associated with them that were just never seen with cannabis or THC exposure. And a lot of this is due to cardiovascular events, again, telling us that synthetic cannabinoids are doing something very different at the CB1 receptor than THC is. And so in my mind, I find it very difficult to look at data generated from synthetic cannabinoids and think that it's going to necessarily be representative of effects that would be seen with cannabis. And I think, especially if we look at the stuff that Parnell's lab had done from UNC that I mentioned before about sonic hedgehog, they indeed found different effects. Whereas when they gave synthetic cannabinoids, they were able to see these effects on sonic hedgehog and even craniodysmorphology that you just don't see with THC. So I have difficulty to begin with thinking that that is going to be hugely informative for us on the human side of things. So we need to be focusing explicitly on THC, I think, if we're modeling cannabis and we're talking about the effects of cannabis. The second point I want to draw attention to is the pharmacokinetics of the model. And I think this is something that, again, wasn't hugely appreciated in the cannabinoid field until recently. And again, something I'm guilty of myself, having done approaches before that I question what the relevance of some of the data might be at this point now. Ideally, if what we're trying to do is match things that are occurring in humans, we want to try and keep the pharmacokinetics somewhat similar so that the levels of fetal exposure to THC are going to be roughly comparable to what we see in humans. Now, it's very difficult for us to actually understand how much THC is getting into the fetal brain in humans because there's been virtually no work in this capacity, but we can start to make some predictions about things, and I'll go through some of the animal data that we've been working on recently. And in line with this pharmacokinetics thing, this really brings about a question that I think Lonnie had alluded to earlier in her talk, which was about how route of administration can impact this and the importance of route of administration. And the reality is, there are pros and cons that exist for each of these models. And so, I mean, the most common typical one, and one historically that's been used in the field the most, is the injection-based approach. And this has obvious pros associated with it in the sense of you're injecting a dose, so you have a fair amount of confidence about the dose that's getting delivered biologically into the system. From the perspective of the researcher who's doing the exposure paradigms, injections are relatively straightforward, and although they can be cumbersome, there's no doubt about that, it can usually be managed in a relatively quick time delimited fashion. So it allows for expediency, and it allows for easier experimental design control, I would argue. Another approach is the edibles approach. This interestingly hasn't been taken up nearly as much in the preclinical world, which is surprising because it actually gets around a lot of the confounds associated with both the injection and, as I'll talk about in a second, the inhalation routes, which have some degree of a stress component associated with them, which, as Lonnie had mentioned, she had a control group to account for that, and is a very important thing to consider because the edible approach you can do in a very non-invasive manner. I mean, there are multiple labs, Nephi Stella and some of the labs at Washington State have put it in like a jello and gotten animals to eat it. Our colleague Stephanie Borglund has done a fair amount with an edible approach, putting it in kind of a, I believe it's a nut butter vehicle that she's giving them, and the animals eat it up. And we've worked with them to show it has really good, you can get good THC into the system through this route. So it is another way. And again, because it's a route of administration used by humans, I think it does have some relevance. And then finally, the inhalation route, which from a pharmacokinetic perspective is one that we believe has a lot of validity in the sense that despite the fact that people do use edibles somewhat regularly, without question, the primary route of cannabis use in humans requires inhalation, whether it be vaping or smoking. It's somewhere between 80 to 90% of people use that route of administration. Some may also use edibles in addition to that, but the overwhelming majority of people who are consuming cannabis do so through inhalation routes. And so there is some effects of inhalation that need to be considered here. Now, the downsides of this, as my amazing grad student, Sam Baglow, has learned, is that it is an incredibly time-intensive and difficult thing to establish. And it's not something that I think the majority of labs would like to take on because it is a very onerous task. And it has proven to have issues associated with it that everyone has to figure out on their own, that we have, largely Sam, has had to troubleshoot on her own in many ways. But at the end of the day, we've got the model working in a relatively stable and consistent manner that we're happy with. And we've done a fair amount of work on the pharmacokinetics of this. And even we just had a paper recently just in adult animals comparing the effects of injected versus vaporized. And while I'm not going to go into it, one of the main ones that came out of it that has brought the potential effects of route of administration to my mind is the difference we see in the production of THC and this bioactive metabolite 11-hydroxy-THC. And the reason I want to highlight this is typically, we only consider THC. When people do the majority of quantification of measurements of the dosing regimen they're giving, they quantify it by looking at what blood levels of THC are like. And this is interesting because 11-hydroxy historically used to be believed to be the bioactive component of cannabis. People thought THC itself may have been inert and it needed to be metabolized into 11-hydroxy before it became active. We know 11-hydroxy seems to, for some reason, accumulate in nervous tissue more than THC itself does. At a biochemical level, we know that 11-hydroxy is at least as potent of an agonist at CB1 as THC. Some work suggest it may actually be more potent. And so if we're not considering what is happening with 11-hydroxy, we're actually missing a large portion of the picture here, because if both THC and 11-hydroxy combined are acting at CB1 to produce these effects, then we need to make sure that we're understanding this. And the reason why this becomes a big thing to consider is because 11-hydroxy is predominantly produced through hepatic metabolism of THC. And one thing we understand about route of administration differences is when THC is inhaled and it goes through a pulmonary route, what we end up seeing is a large reservoir of THC accumulating in the brain and very little production of 11-hydroxy. Whereas when we inject, what we end up seeing is because a lot of the THC accumulates in the adipose tissue and slowly seeps out, it goes through the hepatic route before it hits the brain. And so what we end up seeing is actually more 11-hydroxy being produced than the THC itself. And so we just need to make sure we consider all these things when we start interpreting the data and understanding what it means. And so when we look at the study that Sam has been doing, and this was just published recently, it was a collaborative project with Peg McCarthy's lab, and we did the vaping arm on our half, and Peg was doing an injection study that's actually very similar to Lonnie's approach, which was 5 mg per kg a day for 7 days. So I believe it was actually the same administration paradigm you guys were using, although their window was slightly different because I think they did late gestation. But what's interesting here is, so if we can orient to the figure here, on the left we have THC and on the right we have 11-hydroxy. And then looking at the different tissue depots, we see this is maternal blood we're looking at, this is placenta, and this is the fetal brain. The panel on the left is 15 minutes. So this is immediately following a vape session where the animals were exposed to a series of vape deliveries where they inhaled it, clearly got high. Then we took the moms out, collected maternal blood, collected placenta, and collected fetal brain. We also waited 6 hours and did the same thing. And this is after a 14-day exposure period where the exposures actually started on gestational day 7 and went right to what would have been the end of pregnancy at gestational day 21. And so what we see here is that the maternal blood levels are giving us around between 50 and 75 nanograms per mil of THC. This is roughly approximate to what we would see on kind of the low end of human exposure. So this is totally within the range of blood levels of THC seen following human use of a typical bout of cannabis consumption through a vaporized or an inhaled route. Interestingly, what we see is that there is more THC proportionately in the placenta than what we see in the maternal blood. And while we do see THC getting into the fetal brain pretty readily, it's at a much lower level. So there is some gatekeeper function, let's say here, of the placenta where there does seem to be a lot of accumulation of THC here. And that was something we may have predicted to some degree, but probably not to the extent that it was. But what's interesting is when we actually go and look at 11-hydroxy, what we're seeing is that its levels in the maternal blood are actually quite low. And unlike the fetal, unlike THC at least, we're seeing that we're not seeing this differential effect between placenta and fetal brain, is in the sense that both seem to be getting a fair amount of exposure of 11-hydroxy. And so again, this may go back to the what I'd mentioned before, which is what we don't totally understand, which is for some reason 11-hydroxy really likes nervous tissue and it seems to accumulate there. Some of that's probably the fattiness of it, but THC is also a lipid molecule, and just the addition of the hydroxy group seems to really influence where it's being absorbed and accumulating. So that is, first of all, one thing that's interesting. I think the other thing that's important to point out here is, so this is 6 hours following a vape session, the 14th vape session an animal's had. So the first thing that we can conclude from this is, while yes, we're getting these acute effects, it really does not look like in any real way that we're seeing robust accumulation and storage of THC over long terms, despite the fact that this is after 14 days of exposure. So we weren't sure what to predict about that. Interestingly, the fetal brain actually seems to be the area where there might be a little bit more residue of THC sticking around. And that's possibly because of the fact that a lot of the enzymes involved in metabolism and clearance just don't exist there. And so once it gets in, it may not come out that easily. And so there does seem to be a little bit of residual there. But again, we're talking about incredibly low levels of THC. This is virtually at detection threshold for our assays. So then when we look at injection against this, now again, these paradigms are not comparable. And that's important to note, because one thing we have learned, it is just impossible to create a comparable paradigm of inhalation and injection because of the biodistribution, because of the metabolism. It's just not possible. So we have to recognize that off the bat. We also need to consider the fact that this is a slightly different time point and a different injection duration. This was only 7 days. And this is 3 hours after the injection. But so the first thing that we can notice, and again, this is using a 5 mg per kg dose of THC, is the levels of THC are incredibly higher than what we're seeing from the vape model. So we're already getting pretty high levels of THC in the maternal blood. Again, interestingly, regardless of the approach or any of this, we're seeing a very robust accumulation in the placenta. So that's an interesting internal control almost, or a consistency in the findings that I think really needs to be followed up on, and I'll swing back to this again later. But then I think what's more fascinating about this, and again, this goes back to the differences of an injection route versus a vape route, which is the increase in this production of a bioactive metabolite, which is 11-hydroxy, which had no one ever measured, we would never have seen this. But wow, do we see a big difference in the accumulation in the fetal brain of 11-hydroxy from an injection route versus an inhaled route? I mean, if we look at the numbers alone, we're talking about somewhere between a 10 and 15-fold increase in the levels of 11-hydroxy that we're seeing here. And so again, this doesn't surprise me too much, and it doesn't necessarily mean that the injection routes, that there's better or worse aspects of inhalation versus injection. But the argument that I will always make to everyone is this needs to be considered. We need to recognize that these are not superimposable models. They are producing very different biochemical effects, and the levels of actual cannabinoid, bioactive cannabinoid, that's going to activate CB1 receptors in the fetal brain or in the placenta is different. It is fundamentally different between these. And so we need to recognize that when we have discussions about our translational implications of the data we've generated and what we think it means for a human population. So one thing that was interesting is also using these two approaches, we actually looked at the impact on fetal endocannabinoid levels. So in the stuff that Sam had done with inhalation, we had just had whole fetal brains that we extracted endocannabinoids from and measured. From the stuff Peg gave us, they actually split the brains in half. So we're able to do correlations within the same animal, where one half of the brain we measured endos, and the other half of the brain we measured THC. The lipid extraction and mass spec protocols for these two molecules are wildly different, so we could not do it in the same sample. So, but because they had split their brains in half, we were able to do it with them. So the first point is, quite to our surprise, THC administration had virtually no impact whatsoever on endocannabinoid levels at a gross level. And this was true for the inhalation or the injection model. So despite the magnitudes of difference in terms of how much 11-hydroxy and THC seemed to be getting in the fetal brain from the injection approach, still didn't have any impact. The one thing that came out that was interesting is there was this interesting correlation where the more THC that was in the brain, the less 2-AG. So possibly on an individual variance level, there is an impact of THC to be reducing 2-AG or alternately increasing anandamide. But again, it's difficult to say because we're just not seeing this on the overall analysis level. This is something I think we need to look at further going down the road, is perhaps it's disrupting this natural balance that exists between these two molecules. And what the implications of that mean, we don't know. But that's just something to consider as we move forward. So now Sam's been doing some more detailed work where we actually use the vape chambers to look at the impact of full gestational exposure, starting at G0.5, which is once the sperm plug is identified in the female. So we can assume that's the initiation of pregnancy. And then they begin undergoing chronic exposure daily to these vape chambers to get themselves high on a once-a-day basis. And then we chose 3 different time points, which were G15.5, 17.5, and 19.5. I'm not going to go into depth, but obviously it was because we're trying to capture various discrete windows of specific neurodevelopmental processes, looking at things like cell proliferation and infiltration of microglia and a few other things that we're curious about that cannabinoids might have ability to influence in this gestational period. And so Sam took these tissues, again, we took maternal blood and spleen, we took placenta and fetal brain 4 hours after the last exposure on each of these 3 time points. So looking at the maternal effects of this treatment regimen, so the first thing we know is it is biologically relevant. Exposure to THC is doing something. We are seeing across all 3 of the cohorts, this was data based on their weights on the day that they were sacked. There is clearly a main effect of THC such that there is a somewhat of a slowing of maternal weight gain. It's not a massive effect, but it's definitely there. Interestingly, it doesn't seem to be driven entirely by reductions in food consumption because we didn't see any significant reductions in food consumption. This is true with some other work that's been done that our colleague Ryan McLaughlin, who we've worked a lot with on this vape model, has found that even in adult rodents, they get exposed to vape, they may have like a bit of a munchies response at first. They tend to eat a little less afterwards, so their overall food consumption doesn't really differ, but they seem to have some alterations in energy expenditure and metabolism. And so there could be differential effects on maternal metabolism that THC is inducing that are separate from food consumption that are dictating this and impairing weight gain. Also, interestingly, we reliably saw this reduction in maternal spleen weight from dams that had been exposed to THC vapor. Now, this could be viewed as perhaps a reflection of a low-grade stress response, or it could have some impact on maternal immune function, which again could have some impact on the fetus. And this is something that we're following up as well. But the fact that we see these weight gains and spleen effects tells us that yes, indeed, the exposure regimen we're doing, while not as sledgehammer as doing some of the other approaches with high dose agonist exposure, we still are seeing a very robust effect on maternal biology, telling us that this is doing something. When we look at pup weight, well, we do see this this expected difference in age of the pup, increasing weight gain. We actually didn't see any effect on the pup weight. Now, granted, we only went to P or E 19.5, and I'm going to come back to this in a second, because you can see there's a little bit of a tick down that's starting here. That will become relevant possibly afterwards. We did see reduced litter size across the board as a main effect of THC from the dams that were exposed to THC. So again, we are seeing some biological effects. But again, we didn't really see any effects on pup length. So the pups themselves don't seem to really be that different. Now, this is interesting because work from Ryan McLaughlin's lab that was published where they've done this, they did actually find that on postnatal day 1, similar to the human studies, that they did see this reduced birth weight. So they did find the lower birth weight associated with prenatal cannabis exposure. And again, they didn't see any effects on a lot of other measures like gestation length and other aspects. So this was interesting because they're now looking at postnatal day 1, whereas here we're at 19.5. And so we know this is a period of massive amounts of growth. And so our thought is, at least, we haven't carried through to actually, Sam's doing the cohorts where we let them give birth and grow up now. So we haven't done this ourselves, but our prediction is that we're starting to see some impact here on pup weight, that at that last final surge of growth that's occurring right before parturition, is that where these effects might kick in. And again, because of the increased energetic demands for the fetal growth and the pup growth in that window, and because we're seeing these drops in maternal weight gain, there may be some energetic imbalances that are occurring from this maternal cannabis exposure that are having an effect right at that final window of growth spurt that's happening in the pups. Now, what's interesting about the data that Lonnie had showing that they actually had increased weight postnatally when she was tracking them is this has been seen by other groups as well. I haven't seen anyone track it as early as you guys had, Lonnie, but I've seen data from others. Even Stephanie Borgland's done this and looked, and she's also found from her prenatal exposure that they do see what I would almost call a rebound effect, where there may be low birth weight, but then there's this rebound effect that happens in the postnatal period where they almost gain more weight. So again, something is going on here from an energetics perspective and metabolism, and it's something that we need to understand more, because again, that could have a mechanistic explanation for any of the other effects that get detected from prenatal cannabis exposure. Sam again did the full endocannabinoid analysis to see what was going on, and consistent with previous research, we did find that fetal endocannabinoid levels of both anandamide and 2-AG did increase in an age-dependent manner. But similar to our previous effect, we again found absolutely no effect of prenatal cannabis exposure on endocannabinoid levels in the fetal brain. So this the endocannabinoid levels themselves do seem to be relatively resilient to perturbation by THC itself. Sam also did a bunch of PCR in the fetal brain to look at things like CB1. We also looked at GR. Obviously, we have some stress-related interests here as well, but also because of the effects on the spleen, we were curious about this. And similar to the endocannabinoids, we see nothing on the transcription of CB1 levels, so we're not seeing alterations in that. Obviously, binding is a better measure than mRNA, but it's very, very difficult to do CB1 binding in fetal tissue because of the expression rates of the receptor is much lower than what we see in the adult brain. And we saw no change in GR-mRNA. So we're really not seeing massive effects in some of the systems that we would have necessarily predicted to. But what I can say relatively confidently in our hands is that vaporized, so these are cannabis extract products that are rich in THC. It's predominantly THC, but it is actually a cannabis extract, similar to what Ryan McLaughlin uses. So there are other components of cannabis in here. So we have not been able to find any real detectable impact on the developing endocannabinoid system. We have found some impacts on maternal weight gain and energy balance, and possibly immune function based on the spleen data, which we're following up right now by actually looking at cytokines. We have seen reduced litter size and reduced body weight, which is consistent with the human data. So in general, with the limited data that we see in the humans, our model does seem to generally replicate a lot of the core findings in humans, which does give us confidence moving forward. The pharmacokinetic data indicates that while THC does accumulate in the fetal brain, the placenta does seem to be this primary reservoir where THC accumulates. And this is something that we have to pursue more. And I am not in any way a placentalologist or whatever the word would be for the person who studies this, but Yasmin Herzlab just had a really interesting paper come out recently as well, talking about some placental effects of prenatal cannabis exposure. And in many ways, the placenta is the gate to the fetus. And there is a need for us to redirect some of our attention to the impacts that are happening on the placenta, because I think that that could be the meat of the action for some of what we're seeing here, especially on fetal growth and whatnot. So, I mean, I'm just going to kind of end then by saying, well, what are some of the sustained effects of prenatal cannabis? Now, again, as I mentioned, a lot of the animal stuff is difficult because there are differences in the agonists that are used and the durations that are being administered and a lot of these outcome measures. So I'm just going to highlight 2 data points that have come from some of the preclinical work that I find really interesting. The first is a finding that came from Ryan McLaughlin's lab, and this is again using the vapor model. And what they found was that their prenatal cannabis exposure reduced social play in males specifically, relative to both the vehicle and the air control, because they had a full control system, well, like what Lonnie had, just in case there was stress associated with the air delivery, which they found did account for some of their effects. So I find this particularly interesting because, for one, it's a sex-specific effect in males, and 2, while I'm not someone who's going to say that differences in social play equate to an animal model of autism, I do think that it's interesting that while behavioral changes in some of the other domains like affective behavior and cognitive behavior can be a little dicey to find effects, it's interesting that the effect that a lot of people have seen, because others have seen this as well, is this specificity towards social behavior. And given what we know about the relationship and altered social behavior and autism spectrum disorders, I think, and again, the male specific effects here, I think this is an interesting thing given the human association that was found in the Toronto study that I mentioned earlier. So, and again, we're talking about neurodevelopment in males. So I think that this is something that we need to start paying a little bit more attention to. And I highlight the second paper, and this one was done through an injection-based approach of administering THC from Miriam Mellis's lab in collaboration with Joe Cheer and Ishvan Katona. But they actually found that prenatal exposure to THC produced this really interesting effect on hyperdopaminergic phenotype, but again, only in males, not females. And I find this again interesting because, again, I'm not equating hyperdopaminergic states to psychosis or schizophrenia, but given that there was this effect that has emerged from the ABCD study about this relationship to psychosis proneness in prenatal exposure to cannabis, I find it interesting that we're finding this disturbance in the dopamine system. And the other reason I find this very interesting is because I know Stephanie Borgland's lab has replicated this effect largely using an edibles approach in her animals. And so again, it does seem to be a pretty reliable effect. And again, what this may relate to in terms of a disease state and specificity to males seems like something that needs to be chased in more depth. So just to summarize as I finish, what I've talked to you about today really is the idea that we know that the endocannabinoid system itself is involved in a host of neurodevelopmental processes, especially during in utero development. We know that THC interacts with the endocannabinoid system and thus could impact normative function of the system to modulate the appropriate manner in which neural circuit formation occurs through endocannabinoids acting as axon guidance cues, and thus could also influence behavioral processes or sensitivities to certain diseases that are associated with the circuits that are being perturbed. The human studies, well, as limited as they are, do suggest that prenatal exposure to cannabis can reduce fetal growth. That seems to be the most reliable effect that we've seen. And some of the more recent studies have started to suggest that there could be some association with increased risk of neurodevelopmental diseases that often present as male specific, such as autism or to some degree schizophrenia. The rodent work, I think, can be utilized to examine the impacts of cannabis on the developing brain. And some of the preclinical recent work that's come out has actually, as I mentioned, found alterations in dopamine transmission and social behavior that are male specific, that could relate to the human findings of the relationships between these neurodevelopmental diseases like schizophrenia and ASD that have been started to, started to be linked to prenatal cannabis exposure. So that is all. Again, I want to give a big shout out to Sam Baglow in my lab, who has been a champion in pioneering all the vape work and has, I was listening to some of the comments Lonnie was making in conversations in the break about the difficulties in running some of these studies. And I'm sure Sam would be happy to join in with you about how much work this has been on her and how much she's invested in it. I think it's important to recognize those contributions, especially for how much hard work Sam has done. So. Then the rest of the lab is there. I want to thank everyone for their time and attention. I'm happy to take any questions.
Wow. thank you so much. That was an incredible, incredible talk. I feel like you just gave a very both deep but also broad overview of everything that I've been reading for the past 2 years in an hour. So that was really, really, I know I'm not the only one here.
Sorry I went on so long. I know I lose track of time a little bit.
No, not at all. I really appreciated everything you said, especially directing our attention towards Sam's work as well, because I saw that was just published December of last year and is so hot off the press and so important to this discussion. So I appreciate the fact that it is 3:30 Montreal time. So if anybody does need to leave at once, of course, thank you for your attendance. But perhaps we could take at least one question. It feels like a disservice.
I'm happy to stay for a few minutes and chat.
Thanks.
I've got some time, so I'm flexible.
That.
Hi, Dominique.
I know that there was a question from Alice in the chat. Did you want me to read that?
Oh, sure. Yeah, I can deal with that one first.
Yeah, so I actually wanted to bring something up. So because you were showing the rates for from cannabis use as well as the birth, it was highest for like starting from 15 years old. So basically teenage mothers. And so teenage mothers are usually the ones who have lower, their infants have lower weight during birth, after birth. And so that's one of the things I was wondering about, that there's this, subtle effect of weight, but I was wondering the contribution actually, or the effect of coming from the mother's age actually, that could be playing a role, especially in humans.
Yeah, I mean, the data aren't super well controlled, and they are binned in kind of a wide age range bin, the way they've assembled them. I don't think, I mean, I know that they have relatively, they've tried to create somewhat of matched controls, like in the sense of how they're extracting it. So I know that they're comparing it to women of the same age when they look at these effects, but I don't know if they've aggregated, because a lot of this data has come from different studies. So I would imagine from the way the National Academy did their analysis is because they did a very careful statistical control for things when they essentially did a meta-analysis in and of themselves on how they did this. I imagine they would have controlled for that variable, because I never heard age being the influencing. The only one that I've heard reliably that could be the influencing variable is the co-use of tobacco. And when you control for that variable, that does seem to dilute the effect somewhat. And that's not surprising, because there are effects that we know about in utero nicotine exposure and growth effects. So it could be somewhat contributed to by that. But from what I understand, the comparisons that are being made are within comparable age cohorts. So I would think that that would have controlled for that aspect of it.
Yeah, I'm guessing. And just another quick question in terms of the contribution coming from mothers, but I'm studying microRNAs, and there's some evidence coming from the fact that some epigenetic markers can be transmitted even through semen. And so there's some evidence where fathers who have undergone through severe stress in early life or throughout their life, that actually gets passed on through certain epigenetic markers to their offspring. So I was curious about this because the data you present is very much focused on the mothers and the pregnancy and in this period, but I'm curious about the other side as well.
Yeah, so I can't speak to it because I'm not an epigenetics person. I would direct you to Yasmin Hurd's work. Yasmin has done, I would say, the overwhelming majority of the epigenetic stuff. I know Yasmin has found transgenerational effects that appear to be epigenetic transmitted. I'm going to try to stretch my memory here, but I'm pretty sure Yasmin has shown that she can expose adolescent males to THC and then let them grow up and reproduce with a female, and that there are effects that carry on in the offspring that were linked to epigenetic changes in the paternal sperm. I can't remember what the epigenetic changes were on, or my accuracy of whether that was in the first generation or the second generation that she saw the transmission go for. But Yasmin is kind of I would say she's the queen of the epigenetic stuff in that context. And she's really the one looking at a lot of the molecular changes from the prenatal cannabinoid exposure. So I think her papers would do a much better job of answering that than anything I can do.
Great, thank you very much.
Dr. Walker, do you have a question as well?
Yes, I do. Thank you, Matt. Excellent talk, of course. Hi. Quick questions. When you measure the THC in the placenta, do you know if it was a placenta for male or female fetuses? Or did you just pull them?
I'm pretty sure Sam pulled them. I know Sam's on the call, so she is welcome to chime in if she wants to. I know Sam went through the very laborious aspect of genotyping the fetal samples after the fact. So for being male or female, because we obviously couldn't sex them when they were like E15, it wasn't possible. So we had to do the genetics on it, and took some time. I don't know if we went back and looked at the placenta on that. I know that we did, from talking with Peg, look at different points along the uterine horn, because we know that the placement of the pup can influence the transmission of some substances. And while it wasn't presented there, we didn't find any effect of the uterine placement of the pup in terms of the effects of accumulation. But I don't believe that we found anything that was sex specific from the placenta side.
Mm-hmm.
Do you want me to jump in?
Yeah, Sam, oh, sorry, Sam's there. Sam, if you want to jump in.
Yeah, so the maternal fetal transmission stuff, like Matt was saying, we just have uterine horn and there was no difference. But the effects on litter and endocannabinoids, I do have it aggregated by sex. The dots on the graph technically denote the two different sexes, but there are no differences. So we just have compiled them together on the graphs. Going forward, all of our measures will have both male and female. We just haven't seen any sex differences up to this point.
And I think, Sam, did Peg sex her animals? Because I feel like they may have. And they
Yeah,
didn't see
she did in the
yeah.
maternal fetal paper.
Yeah.
They also didn't see any differences.
So yeah, with Peg stuff, we didn't see any sex differences on the injection model side.
And I was wondering, your THC extract, do you control for all the entourage effects of the terpenes and all those other phytocannabinoids that can be present, or?
Yeah, I mean, ours honestly is, I mean, I believe the batch of THC that we got from Afria when Sam did this was 96% THC. And from that, we dilute it down to basically a 10%. So we make it down to 100 mgs per kit, or 100 mgs per mil. So if I remember, the only other cannabinoid that we've really seen at any level that would possibly have biological effects was CBG, or cannabigerol. Which could have some effects, but I think it was maybe 1%. And that was pre-dilution, which would mean that in the final volume of what was vaped, it was about 0.1%. So I would be quite surprised if there was enough CBG or anything else in there to actually have an interactive effect. Ryan, what Ryan works with for his prenatal stuff and all his vape stuff is he has an extract that NIDA provides him. It is, he gets some analytics with it as well, and it can vary, and he has to do an extraction because he gets sent something that's basically the equivalent of tar. So he has to do an ethanol extraction to pull all the lipids out and redo a THC suspension from that. And he gets both a high THC and a high CBD one. And his have a little bit more of the cannabis components in it. But again, his effects don't seem to vary that much. I mean, I'm of the position that the entourage effect could do something, but I'm going to be a little reluctant on believing that until I actually see data to suggest that there is a genuine effect of an entourage effect from these other molecules, because some of it could be present. I think it really depends on the concentration that it's present in. We have gotten now a whole cannabis extract that has everything in it. It is still enriched in THC, but it has the whole plant with it. That we're starting to do vaping with, and at least in our other models, we really haven't seen gross differences in the effects that we've measured with it. So it's certainly possible, but we try and do all our studies with one batch of extract that we get, so that at least it's internally consistent with itself, and we have the analytics of that batch that we get. But these are really like the stuff that Sam's done so far. Really it's predominantly THC. There's just little bits of other stuff in there, but it's at pretty dilute levels.
Thank you. And just one more comment is, I wonder if the change in body weight after birth could be related to the suckling, because endocannabinoids are so necessary for suckling that you may have some changes here that account for the change in weight postnatally.
Yeah, and I mean, the other thing that we've got to do, and I've talked to others who've done this, is I'm also curious because technically the mom's going to go through withdrawal to some degree. Now, obviously, the nature of cannabis dependence and the way that they have withdrawal syndrome is fundamentally different than a lot of other drugs. And from talking to others, I mean, Sam's going to be scoring maternal behavior because we haven't had a cohort that's given birth and tracked. but she's going to be monitoring the maternal behavior to see if we can see anything. Mostly everyone else has not that I've talked to, because that was the other thing I was wondering is like, is this like kind of the stuff that you and Ryan had done when he was in your lab with the stress model, where some of it seems to be the transitory nature of the mumps, so the pups don't latch on long enough to trigger the milk ejection.
But maybe you can just record the milk ejection reflex at the same time.
Yeah, no, I definitely think there could be something there. I mean, a lot of the weight is, at least the human stuff is certainly found in the newborn as it comes out, that they see the weight difference. And because in the stuff Sam had done, we only went to 19.5, there was still about a day and a half of growth left. And because we'd started to see it, but Ryan saw it in the first day after birth. And as Lonnie has seen, there's almost a rebound that seems to kick in a little bit afterwards. I'm curious, and that's why I'm not sure. I kind of wonder whatever THC is doing to maternal metabolism and kind of the bioenergetics of that energetic demand that's required right at the end of pregnancy, especially if that's having an influence on what, nutrient availability or whatever is able to get to the fetus so that it has that last growth surge right before birth. And maybe it's just not quite enough, but I don't doubt that there's also going to be some degree of postnatal effects.
Thank you.
Great, thanks so much. Millar, do you want to ask a last question and then we'll close out for the day?
Sure. Thanks so much, Matt. What a wonderful talk. Really incredible breadth of stuff you covered in a very short period of time. So thanks so much. One quick question. One thing that didn't come up in your talk was THC to CBD ratio and how that may impact, any of the phenotypes that you mentioned today. So do you have any thoughts on that and where the data is on that?
So first of all, I would just say my general opinion is that I think there's a little too much emphasis put on CBD. And I say that because the reality is CBD doesn't really exist in street cannabis, in any form of recreational cannabis. It just doesn't exist because THC and CBD compete for biosynthesis. They use a common precursor. So as soon as cannabis was bred to predominantly make THC, it did so by essentially enriching the enzymes that convert the precursor into THC and not CBD. And so what you end up finding is that in virtually every cannabis that exists on the street and on the market that people buy recreationally, unless it's intentionally bred for CBD, there is virtually no CBD, and it's always under 1% of CBD that's in there. So I don't know how reflective that is of anyone who's using normative cannabis, because CBD is just not a molecule that's ever there. The only reason it would be relevant, I would assume, is that there is clearly a subset of people who use CBD exclusively because they don't want to become intoxicated from THC. So I'm not sure how important the ratio of the two of them would be, because there is essentially no ratio that exists in real cannabis. But I do think there's a huge importance of looking at the effects of CBD in and of themselves, because a lot of women are using that as well. For one, probably more than THC, because there is this general belief that CBD is harmless and it only provides medical benefit or health benefit in some capacity. And so they don't think that there's any harm associated with that. And so you're going to get people who are taking it. But the reality is we know even less. And actually the small amount of studies that have come out with CBD, there's been some work that's come out of Edmonton, actually, that's been looking in kind of a zebrafish model. And they actually find very significant and robust teratogenic effects of CBD exposure. And again, we're not clear why, because this is not through a CB1 mechanism, because CBD isn't an agonist at CB1. We're not sure what it's doing, but there has been a couple of signals that I've seen at some cannabinoid meetings as well that have suggested that there may be effects of CBD that people are just not considering. So I would also push for the field to actually look at CBD in isolation a lot more, because I think it's important to understand this, because one of the things that we've learned from the clinical trials of CBD in humans is that CBD exists in this, I don't know, mythological realm, let's say. Everyone seems to think it cures everything, when reality, there's certainly some things, pediatric epilepsy, there's some other effects. Yasmin's had some interesting effects in opiate craving. In the clinical world, there is some effects of CBD, but you never see them unless people go at doses of 300 mgs up to 2000 mgs a day. Like, they're very high doses of CBD that are required. There's never been a clinical study that I've seen that's been run in a blinded, proper controlled fashion that has ever found an effective CBD under 300 mgs. So the majority of people who take CBD do so in what I would refer to as CBD homeopathy, because they take super low doses of CBD. They're convinced that it's doing biological effects to them, when the reality is we know it doesn't really leave the gut at all. So like when you're taking 20 mgs of CBD, you're really just essentially making highly expensive human excrement, let's say, because not a lot of it is leaving your GI tract. So I wouldn't be surprised at that. But if people actually start taking CBD at doses that are used in clinical trials, the problem is it starts interacting with liver enzymes. So it's not innocuous. And you'd probably see the same thing with THC, but the doses of THC that would hit those enzymes and saturate them, you would never approximate because the person would become so overwhelmingly intoxicated that it would never hit that level. But we've learned from the pediatric epilepsy studies that the high doses definitely saturate liver enzymes and cause accumulation of other substances. And there was even a study done in healthy humans that just gave a dose in that range. I think it was the high hundreds or maybe 1,000 mgs to an adult, and found even a week of dosing adults at a dose that's clinical, they started to see changes in ALT and hepatic enzymes that are suggestive of liver inflammation or liver damage. So the idea, like the mythology that surrounds CBD, I mean, it's one of these things that will trigger a massive rant in me, just because there's so little data that supports any real benefit of it. People take it like gangbusters and swear by it in many ways. And the reality is, I would actually be concerned, because of the doses that you need to take it for any kind of therapeutic benefit, that there are going to be biological effects. And so if pregnant moms were taking it at that level, I would be very concerned. Because again, we have no idea what the accumulation is doing. We don't really even understand the biological mechanisms of CBD, because like I said, it's not a cannabinoid agonist. It interacts with the adenosine systems and probably serotonin systems and a few others, but even the pharmacology of CBD is a mess. So no one knows what it's doing, but it's going to certainly accumulate in lipid-rich environments. There's no question on that. So I would be very curious to see what the effects of CBD on developing fetuses are. And it's probably not something that we're going to pursue at all, just because it's a huge amount of work in addition to what we're already starting. But it is one of the things that I hope other people start looking at more, because I think it's important for us to get this information and understand, is it actually safe or not? Because we're focusing very heavily on cannabis, and we're kind of deluding ourselves into thinking that CBD is this like super healthy, awesome, like miracle drug that has no detrimental effects.
Wow, that's a fascinating perspective, and one that I haven't heard expressed in quite that way. I really appreciate that, and your perspective overall, because it's clear that you have a great like overarching view of the field, where it's at, next steps of where we can all take it as well. So thank you so much. Thank you for your presentation and moderating the trainee session, and your time and staying overtime for that. We all really appreciate that. And thank you all for your participation today. I look forward to seeing you tomorrow for our final day of the conference. With that, I'll say goodbye. Thanks.